Dear learners, in this video, we are going to discuss about uptake of nutrients by the cells. Materials move across the plasma membrane of both prokaryotes and eukaryotes by two kind of process. One is passive process or passive transport, and the another one is active process or active transport. So the passive process is a substances which is cross the membrane from an area of high concentration to the area of a low concentration. Here the move with the concentration gradient or a difference without any expenditure of energy, that is ATP by the cell. So the passive process or passive transport, which include um, three different methods. One is simple diffusion, facilitated diffusion, and osmosis. Simple diffusion. Here, the simple diffusion is a net movement of a molecule or ion from an area of a higher concentration to the lower concentration. For example, a dye which is put into a beaker of a water, the molecule of a dye in the pellet diffuse into a water from an area of a higher concentration to the lower concentration. The movement contained until the molecule or ion are eventually distributed. So the point of even distribution, which is also called as equilibrium. So the cell rely on simple diffusion to transport certain molecule like water, oxygen, carbon dioxide across the membrane. Facilitated diffusion. In facilitated diffusion, an integral membrane protein function as a channel or carrier that facilitate the movement of a ion or large molecule across the plasma membrane. Like the integral protein are called a transporter or permeases. So facilitated diffusion is similar to the simple diffusion method. So in that the cell does not expend energy. Why? Because the substances move from the higher concentration to the lower concentration. And this process differ from the simple diffusion in its use of a transporter. Next method is the osmosis, and it is the net movement of a solvent molecule across the selectively permeable membrane from an area with the higher concentration of a solvent molecule which contain the lower solute molecule to an area of a lower concentration of a solvent molecule which contain the higher concentration of a solute molecule. So in the living system, the chief solvent is a water molecule. So the water molecule may pass through plasma membrane by moving through the lipid bilayer. So by the simple diffusion or through the integral membrane protein, which is also called as aquaporin, that function as a water channel. So bacterial cell may subjected to any three kind of osmotic solution. The first one will be isotonic solution. So isotonic solution, which is nothing but the water concentration is equal inside and outside the cell. So thus rate of diffusion are equal in both the directions. So here not net movement of water will be there. In the second solution will be hypotonic solution. Here water move into the cell. So if the cell wall is strong, the cell swells, and if the cell is in the weaker condition, it will get damaged. So the cell burst, which will release the osmotic lysis. So the third solution will be hypertonic solution. So the water move out of the cell and the cell becomes a string, so which is called a plasmolysis. In the active process, the cell must use energy to move substances from area of a low concentration to the area of a high concentration across the plasma membrane. So the substance actively transport or ions, amino acids, and simple sugar. And although these substances can also be moved into cells by passive process, their movement by active process can go against the concentration gradient, allowing a cell to accumulate needed material. ATP binding cascade transporter, which is an example of a active transport system. It is also called as ABC transporter system. It is a very important example of a active transport system. And they are also observed in the bacteria, which is very common in the gram-negative bacteria, arc-bacteria, and eubacteria. In that, the first solute binding to the protein, so which is periplasmic binding protein, which is bind to the substrate to be transported and approach the ABC transport complex. When the solute binding protein attaches to the transporter and release the substrate, which is moved across the membrane with the aid of a ATP hydrolysis. Through translocation, in the active transport, the substance that cross the membrane is not altered by the transport across the membrane. So in the group translocation, a special form of active transport that occur exclusively in the prokaryotes, the substances is chemically altered during the transport across the membrane. Once the substances is altered and inside the cell, the plasma membrane is impermeable to it, so it remains inside the cell. This important mechanism enables cell to accumulate various substances even though they may be in the very low concentration outside the cell. Group translocation require energy supply by high energy phosphate compound such as phosphoenol pyruvic acid. So one example of group translocation is a transport of a sugar glucose and mannitol, which is often used in the growth media for the bacteria. So this phosphoenol pyruvic acid uh, combined with the glucose molecule which produce the pyruvate and glucose 6-phosphate. And the second example is the phosphoenol pyruvate with the mannitol which produce the pyruvate and mannitol 1-phosphate inside the cell. Iron uptake by the microorganism. Almost all microorganisms require iron for the use in cytochromes and many enzymes. So the iron uptake is made difficult by the extreme insolubility of ferric ions and its derivative, which leaves a little free iron available for the transport. Many bacteria and fungi have overcome this difficulty by secreting siderophores. Siderophores are the low molecular weight organic molecule which are able to complex with the ferric ion and it is used to supply the cell. Ferrichrome is a hydroxamate produced by many fungi and enterobacter is the catecholate formed by the Escherichia coli. And it appears that three siderophore group complex with ion to form a 6 coordinate octahedral complexes. So the microorganisms secrete siderophore when iron is scarce in the medium. So the once the iron siderophore complex has reached the cell surface, it binds to a siderophore receptor protein. Then the iron is either released to the cell directly or to the whole iron siderophore complex. It is transported inside by an ABC transport mechanism. In E. coli, the siderophore receptor is the outer membrane of the cell envelope. When the iron reaches the periplasmic space, it moves through the plasma membrane with the aid of the transporter. After the iron has entered the cell, it is reduced to the ferrous form. Iron is so crucial to microorganisms that they may use more than one route of iron uptake to ensure an adequate supply. Thank you.